Hello viewers, I am Lata Adhikari from Lokanayak Omeo College, Dhekiajuli, Assam. First of all, I would like to thank the entire team of Zoological Society of Assam for providing us a platform to help undergraduate students during the lockdown period. In this presentation, I am going to discuss on a very interesting topic called genetic basis of sex determination. Since time immemorial, sex determination has always puzzled as well as fascinated mankind about the differences between sexes. Aristotle suggested that the heat produced during copulation decides the sex of the progeny. More heat produces male and less heat produces female. This may be true in case of reptiles but not applicable to all organisms. Therefore, this presentation is a humble attempt to give an account of various factors which may determine sex. Learning outcome. By the end of this discussion, the learners will gain an insight into what is sex, sex determination, gonadal differentiation, chromosomal theory of sex determination, male-haploidy or haplodiploidy, genic balance theory, environmental and other sex determining factors, effect of autosomal gene on sex or single gene effect, and cytoplasmic sex determination. Now, what is sex? Sex can be defined as an aggregate of morphological, physiological, and behavioral qualities that can differentiate the organisms producing eggs from those producing sperms. Organisms producing eggs are called females and those producing sperms are called males. Sex determining chromosomes follow Mendelian laws of inheritance. Sex determination. It is a process that leads to distinction and separation of structures responsible for production of male and female gametes. Gonadal differentiation. After fertilization, a zygote is usually predestined to develop either into a male or a female. But in birds and mammals, there is an exception. The undifferentiated gonad has two main regions, the central medulla and the peripheral cortex. Here we can see the undifferentiated gonad. The outer part is known as the cortex and the inner part is known as the medulla. In the presence of HY antigen, in this undifferentiating gonad, the cortex regresses and it develops into a testis. Now, if there is no HY antigen, in the differentiating gonad, the medulla regresses to form a ovary. According to HY histocompatibility, Y chromosome-linked antigen hypothesis, the heterogametic sex is dominant in birds and mammals. While homogametic sex is neutral, the presence of HY antigen on the surface of the embryonic gonad cell leads to the development of gonads of the heterogametic sex, that is, mammalian males and avian females. Vice versa happens in the absence of the antigen. Now we will discuss chromosomal theory of sex determination. Before that, some history of the sex determination. In 1891, Henking, a German mathematician, observed X chromosome in some male bugs. He could not define its significance. Normally in mathematics, any unknown variable is depicted by X. As Henking was a German mathematician, so he termed it X chromosome. In 1905, Wilson and Stevens demonstrated that in Protina, both male and female had extra chromosomes beside normal ones. Those were known as supernumerary chromosomes. In 1908, Stevens discovered Y chromosome. In 1909, Wilson gave accurate account of X chromosome. Now chromosomes are of two types, allosomes or sex chromosomes which determine sex, autosomes, body chromosomes, and these chromosomes determine bodily characters. Majority of the diploid sexual animals have a pair of sex chromosomes on the basis of which sex is determined. Usually, females are homomorphic and males are heteromorphic. Besides chromosomes, other factors which determine sex are environmental factors, cytoplasmic factors, etc. Types of chromosomal mechanism of sex determination. First type is sex chromosomes undifferentiated. In primitive forms, sex genes are located in autosomes. In the second, B type, XX or XY lineage type. It was first studied by Wilson and Stevens in lineage turgicus, which has two types, female homogametic, male heterogametic. In the second type, females are heterogametic and males are homogametic. In the first case, we will say females homogametic, that means XX, and males are heterogametic, that means XY. These examples are found in case of Drosophila or man. Here we can see in case of man, females are having 22 autosomes and there are 2 X chromosomes. In case of males, there are 22 autosomes plus XY as because these are heterogametic. Females produce same type of gametes 11A plus X. Males produce two type of gametes 11A plus X, 11A plus Y. So here we can say if it is 22 autosomes plus same type of X gamete, they develop into females, and if it is 22 autosomes plus XY, these are males. Modifications of XX, XY lineage type. First one is X and Y chromosomes attached to a pair of autosomes. In the second type, X or Y complex, X or Y chromosome is broken into fragments. Examples are found in mantis, E. coli insect. Here we can see male has 20 autosomes, 5X plus Y. That means the X chromosome has been fragmented into 5 parts. In case of female, there are 20 autosomes and 5X plus 5X. Male produces two types of gametes, 10A plus 5X, 10A plus Y, and females they produce same type of gametes. When there are 20 autosomes plus 5X plus 5X, it develops into a female, and if there are 20 autosomes 5X plus Y, it develops into a male. In the second case, females are heterogametic and males are homogametic. So females are represented by ZW and the males are represented by ZZ. That means the X has been replaced by Z and Y has been replaced by W. The examples of this type of cases are found in moths, fowls, birds and fishes. Here we can see in female moth there are 54 autosomes and it is ZW. In case of male there are 54 autosomes and they are homogametic with ZZ. So female produces two types of gametes, 27 autosomes plus Z and 27 autosomes plus W. Male moths they produce same type of gametes. So when we get 54 autosomes plus ZZ, they develop into males, and when there are 54 autosomes plus ZW, they develop into female. Third type is the XX female XO proteiner type. McClung, while studying meiosis in testes of squash bug, observed the following. He found that in case of females there are 22 autosomes plus XX, and in males there are 22 autosomes and single X. So females produce same type of gametes, that is 11A plus X, but males produce two type of gametes, 11A plus X, the other is single 11 autosomes with none of the X chromosome. So when there are 22 autosomes plus XX, it develops into a female, and when there are 22 autosomes and a single X, they develop into a male. Fourth type is Hymenoptera type, where females are diploid and males are haploid. In 1850, Dzierzon observed in honeybee, wasp, and etc. In case of honeybee, females are diploid and males are haploid. By the process of meiosis, the female gives rise to haploid set of zygotes, whereas by the process of mitosis, male produces haploid set of zygotes. Here we can see the haploid set of zygote produced by the female, they develop into males, or commonly known as drones. They develop parthenogenetically, and parthenogenesis means development of an egg without fertilization. Now here we can see haploid set is coming from the female part and another haploid set is coming from the male part. They produce the diploid females. If these females are fed with the royal jelly, they develop into queen, which are fertile females. And if they are not fed with the royal jelly, they develop into workers who are sterile females. Now we will discuss about the genic valence theory. In 1916, C.B. Bridges put forward the theory which states that every individual, irrespective of its sex, possesses in its genotype depicting the characteristics for both male and female. Which sex will actually develop is decided by the balance between the autosomes and sex chromosomes in the individual. In Drosophila, genes for maleness are present in autosomes, while for femaleness in X chromosomes. Sex is determined by the ratio given below. Sex ratio is equal to number of X chromosomes by number of autosomal sets, which is shown as X by A. For normal male, X by twice A is equal to 0.5, and for normal female, twice X by twice A is equal to 1. If the ratio is less than 0.5, the individuals develop as super males, which are generally weak, sterile, and not viable. If the ratio is more than 1, then these individuals develop as super females, which are generally weak, sterile, and not viable. And if the ratio is between 0.5 to 1, these individuals develop as intersexes, which are intermediate between male and female with sex expression of both primary and secondary characteristics. Here I have given the table for your ready reference where you can find the sex index ratio for normal female, then normal male, super female, super male, and intersexes. Now we will discuss about the gynandromorphs. Gyne means female, andros means male, and morph means form. Individuals exhibiting the phenotypic characteristics of both male and female are called gynandromorphs. Probably gynandromorphs arise from XX zygotes. During embryonic development, in one or more cells, one of the two chromosomes does not pass to any pole at anaphase, and as a result, one of the daughter cell possesses twice A plus twice X and the other half twice A plus X. Twice A plus twice X cells divide and give rise to the female parts of gynandromorph, while twice A plus X give rise to the male part. If the irregular distribution of X chromosome occurs at the first mitotic division of zygote, the male phenotype would extend to exactly half of the body of the gynandromorph. The extent of male part in a gynandromorph therefore depends on the stage of embryonic development when the irregular distribution of X chromosome occurs. The earlier the occurrence, large the size of the male portion. Example of gynandromorph is found in Hindu mythology, where here we can see the Ardhanarishwar. Here one lateral half is that of the male part Shiva and the other lateral half is that of the Parvati or the female part. In nature, we do find the gynandromorphs in most of the insects, and here we can see the Morphoretinor, which is a butterfly, and here one lateral half is of female and the other lateral half is that of the male. Now we will discuss environmental and other sex determining factors. In some animals, sex is determined by the environmental conditions. For example, in Bonellia, a sea worm, sex is undifferentiated. Free-swimming larvae that settle on bottom develop into females. Females are a meter long with long proboscis. The larvae which settle on proboscis of female develop into males. Males are 1 millimeter, lack digestive tract, have well-developed reproductive organs. They migrate to uterus of females and reside there as parasites. Here we can see the image of the Bonellia, where this is the female and it has a long proboscis, and this is the male. It resides in the reproductive part of the female. And this is the example of our Dinophilus, which is again another sea worm. Here the egg size determines the sex. Small eggs develop into males and larger eggs develop into females. In sea fishes, example labroids, social order of an individual determines sex. Single largest male having largest body is dominant. Rest all individuals are females. Females are all hermaphrodite. Their body size determines their position in school. If male dies, largest female change into male within 14 to 18 days to take his position. The larvae of crepidula in the company of a female develops into a male. These are the images you can see. This one is labroids and this one is crepidula. In some reptiles like lizard Agama agama, if the eggs are incubated at 28 degrees centigrade, 30, 32 or 34 degrees centigrade, they develop into males. And if the eggs are incubated at 26 or 36 degrees Celsius, they develop into females. In turtles, Chrysema picta, if the eggs are incubated at high temperature, that is more than 33 degrees Celsius, they develop into females. And if the eggs are incubated at low temperatures, that means below 28 degrees Celsius, they develop into males. Effect of autosomal gene on sex. In Drosophila melanogaster, Sturtevant discovered a recessive gene, transformer gene or TRA, with no observable effect in heterozygous condition on either sex. However, in homozygous condition, TRA gene transforms female into phenotypic males, that those are sterile. In homozygous condition, it has no effect on males. This happens due to one autosomal gene SOX9. In humans, ideally, XX chromosomes should develop into females, but if they have an extra copy of SOX9 gene, they develop into males even in absence of SRY gene, which is maleness determining gene. Effect of SRY gene or TDF gene, that is testis determining factor on sex. SRY, sex determining region of the Y chromosome, determines male sex in humans. However, its studies in mice has revealed that its major role is to help the expression of its related gene SOX9, which induces Sertoli cells differentiation, which in turn results in testis formation. SRY is a small single exon gene which provides instructions for making a protein called the sex determining region Y protein. It is located in the small arm of male specific region of the Y chromosome. This protein is involved in male sexual development. The protein produced from this gene acts as a transcription factor, which means it binds to specific regions of DNA and helps control the activity of particular genes. This protein starts processes that initiate development of male gonads in fetus. Simultaneously, it inhibits the development of female reproductive structures. Reduced or delayed SRY expression impairs testis development. Moreover, deletion or mutation of SRY genes give rise to XY females with gonadal dysgenesis, who appear to be normal females at birth, but at puberty neither they menstruate nor develop secondary sexual characteristics. They have fibrous gonads without eggs. Testicular feminization gene. The testicular feminization gene is located in X chromosome. XY individuals carrying this gene sometimes may depict feminine characteristics such as breast and vagina and degenerated testes. It happens due to inability of testosterone hormone to express itself. Now we will come to double sex gene or master gene. This gene can express in both male as well as female flies, subject to alternative splicing, producing the protein isoforms DSXF in females and DSXM in males. It also regulates somatic sexual differentiation in both males and females. It is a gene that is involved in the sex determination system of many insects, including Drosophila. In males, the DSX gene represses the gene responsible for female sexual differentiation, so that male differentiation function are expressed. In females, it represses the genes for maleness, so that female sexual differentiation functions. Cytoplasmic sex determination. In bacteria, sexuality is controlled by cytoplasmic factor called fertility factor or F factor, which was found by Lederberg and Tatum in 1947. He found that male or donor cells possessing F positive and female F negative or recipient possessing none. During conjugation, the F negative factor becomes recipient cell, converting them into F positive or the donor cells. This is the figure of transformation of F negative recipient to F positive donor bacteria. Here I have given the references, and these are the some of the suggested readings. And you can give me your feedback. This is my email ID. I thank you all for giving me a patient hearing.